Welcome everyone to Plant Health TV, a global channel focused on the International Day on Plant Health 2020. Are you ready to learn about plant health? Let's start now. My name is Manuel, your TV presenter, a Ph.D. student from the Institute of Sostenible Agriculture. My research is based on the olive microbiome and the role of this microbial community as a biocontrol agent against vascular pathogens like Verticillium dali and Xylella fastidiosa. But you may be thinking, why olive tree? Well, they maintain environmental function in Mediterranean ecosystem, and olive oil is the basic food of the Mediterranean diet. Olive tree, like other living organisms, is colonized by a series of microbial communities that impact on growth, health, and pathogen resistance. To characterize this microbial community, we insert an olive branch into the Scolander chamber device and collect the serum sac, which it will be used for DNA extraction and sequencing. So after the DNA sequencing, what is the next step?
The first step is the quality control of the DNA sequence to eliminate short, low-quality reads and chimeras. And this is the more important step for further analysis.
And how can we identify the name of each microorganism precisely?
Then high-quality sequences are aligned to a reference database where each DNA sequence is associated with a specific microorganism that are in our olive sealant sample.
Now it's time to know a little bit more of one pathogen, Xylella fastidiosa. Can you tell me the main characteristics of this bacterium?
Biofilm formed by Xylella fastidiosa cells occlude xylem vessels and cause several symptoms in crops like olive and almond tree. Also, ornamental and forestry plants.
And how do you isolate this bacterium in order to perform your experiment?
We obtain xylem extract from infected plant tissue, and it is grown in a specific culture media at a constant temperature.
But other questions came to my mind related to Xylella fastidiosa detection. How can we detect directly Xylella fastidiosa?
There are many techniques available in the EPRO protocol, like conventional PCR, qPCR, isothermal PCR, or the most recently, the droplet digital PCR, which use depend on the need.
And how do you differentiate the diversity of this bacterium?
Also with molecular tools, but now I take advantage of Oxford nanopore technologies for selective amplification of DNA to find the different subspecies of Xylella fastidiosa.
Taking everything into account, if we know what microorganisms live in olive tree, we can identify the microorganisms that are involved in the defense of the plant against this pathogen, achieving a greater health and viability of olive cultivar. That's all for today. See you in our next program, and remember, take care of plant health.